Okay, our second learning objective is to look at different organelles, and I'm going to tell you about what the functions of those organelles are. So we'll start with the nucleus, that is the one that's most obvious in a eukaryotic cell. Includes almost all of a cell's genes, and if you look under a microscope, that's the one you're likely to see. So the nuclear envelope is a double layer of membrane material that encloses the DNA and the proteins associated with the DNA and separates it from the cytoplasm and all the stuff that's happening in the cytoplasm. And that nuclear membrane is actually a double membrane. So there's two layers of phospholipid bilayers that separate the DNA from the cytoplasm. So here's a picture showing you all of the things that are going on with the nucleus. You've got the DNA protein complex that's here in the middle. There's an even darker spot in the middle of the nucleus called the nucleolus. Nucleolus means little nucleus. And then you've got the envelope itself, which has two layers of membrane material, an inner membrane and an outer membrane. And then there are these pore complexes, which are proteins that allow for the passage of materials into and out of the nucleus. So you can see that the nucleus, it looks like it's separate from everything else, but it's actually physically attached to some other membrane-bound organelles. This one's called the ER, and I'll tell you about the ER in a minute or two here. So chromosomes make up your DNA, and in a human, typically what you have are 22 pairs of chromosomes and then one pair of sex chromosomes. So in this case, we're looking at the chromosomes of a female because she has two X chromosomes. A male would have an X chromosome and a Y chromosome. And what this picture is showing us is these are chromosomes that have been stained with different fluorescent dyes. So what you can see in this picture is that when the cell is normally doing its thing, it's not dividing, the chromosomes are not condensed into these linear forms. They're in this sort of shape here. They're more diffuse. They're sort of unpacked, and they're all in different regions throughout the nucleus. If they were not in different regions in the nucleus, you would expect this not to be patches of different colors. You'd expect it to be sort of all one kind of grayish blob if all of the DNA from each chromosome was sort of intermingling with each other. But you can see that even in this very colorful image here, that there's discrete regions. There's patches that represent particular chromosomes. So when the cell is not actively dividing, the chromosomes are unpacked as this material called chromatin. And then when the cell is going to divide, all of these regions condense down and get packed up into these segments that we call chromosomes. Ribosomes are where protein is made. And ribosomes are, they're not membrane-bound organelles, but they exist as particles that are made out of RNA molecules, like we talked about in the previous chapter, and proteins. So ribosomes are where proteins are made from working copies of a gene using messenger RNA. Now, we find ribosomes either just floating around within the cytosol as free ribosomes, or sometimes they're attached to this other structure called the endoplasmic reticulum, or on the outside of the nuclear envelope. And we call those bound ribosomes because they're attached. They're bound to another structure. Now, a ribosome, in my mind, it kind of looks sort of like a hamburger bun, where it has a big part, like a top half of the bun, which is the large subunit, and the bottom half, which is the small subunit. And different organisms have different sizes for ribosomes. Prokaryotes have very different sized ribosomal subunits than eukaryotes. And here is I don't know whose feeling I'm going to hurt, GIF or GIF of a ribosome at work. So this structure in here is a messenger RNA molecule. And these dark blue structures are transfer RNAs that are bringing in different amino acids. And this is the polypeptide. This is the protein that is being produced. And we're going to talk about this process in a bit more detail when we are talking about genetics. But I just thought it's a cool thing to show you right now. Just kind of how a ribosome does its job. And in this case, it's attaching to, it's becoming a bound ribosome. It's attaching to the endoplasmic reticulum, and it's going to put that protein inside the space inside the ER. We haven't talked about ER yet, so don't worry if you don't know what that is just yet. And that's it.